Hey, today let's talk about how we can use DNA chips to detect mutations in the breast cancer gene. But first, let's understand what DNA chips are. DNA chips are small devices that have many tiny probes of DNA which can identify specific DNA sequences. These chips are commonly used in research and clinical applications in the field of genomics. DNA chips have many clinical uses, including gene expression analysis, pharmacogenomics, infectious disease diagnosis, and cancer diagnosis and classification. But for today's discussion, we will focus on using DNA chips to detect breast cancer related mutations. So, what does a DNA chip look like in simple terms? Well, it's like a small piece of glass or a silicone chip with a bunch of tiny spots arranged in a grid. Each spot has a very specific DNA probe. DNA probes can be made on DNA chips using a process called photolithography. This process involves creating a pattern of light on a photosensitive material, which is then used to create the DNA probe pattern on the chip. Here is a general overview of the process. First, the chip surface is coated with a photosensitive material. A mask is created with the desired DNA probe pattern. The mask contains transparent areas where light can pass through, and opaque areas where light cannot pass through. The mask is placed over the photosensitive material and exposed to ultraviolet light. The areas of the material that are exposed to light become chemically altered and are able to bind DNA. The mask is removed and the chip is washed to remove any unbound material. DNA fragments, typically about 20 to 30 bases in length, are then synthesized with a specific sequence that is complementary to the target DNA. These fragments are attached to the modified areas on the chip's surface. When the chip is exposed to a sample containing the complementary DNA sequences, these sequences will bind to their corresponding probes on the chip, allowing DNA to be detected. The result is a chip with an array of DNA probes that can be used to detect the presence of specific DNA sequences in a sample. The size and number of spots that contain probes can vary, but most chips can hold thousands to millions of probes. There are also different kinds of DNA chips that can use different ways to show the results, like using one color or two colors. To prepare a DNA chip labeled with fluorescence, a small amount of the sample containing the target DNA sequences is first extracted. Next, the DNA sample is labeled with a fluorescent dye, which can be done using a variety of methods such as Nick translation, random priming, or PCR amplification with fluorescently labeled nucleotides. Once labeled, the DNA sample is then hybridized to the DNA chip, which contains many small spots, each containing a specific DNA probe sequence that is complementary to the target sequence. During hybridization, the labeled DNA sample will bind to the complementary probes on the chip. The chip is then washed to remove any unbound DNA, leaving only the DNA that has hybridized to the probes. To detect the hybridized DNA, a fluorescent scanner is used to measure the intensity of the fluorescent signal emitted by the labeled DNA on the chip. The intensity of the signal at each spot on the chip corresponds to the amount of hybridized DNA, allowing researchers to determine which probes have bound to their target sequences. So then how do we apply DNA chips to detect breast cancer mutations? When it comes to breast cancer, scientists use DNA chips to find out if there are any changes in two genes called BRCA1 and BRCA2. These genes can make it more likely for someone to get breast cancer. Here are the steps to use DNA chips to find out if someone has changes in those genes. First, the doctors collect a small piece of someone's DNA, usually from their blood or spit. Then, they make many copies of the DNA using a process called PCR. Next, they label the DNA with special tags or markers that make it easier to see on the DNA chip. The labeled DNA is then put on a DNA chip that has thousands of probes that match specific parts of the BRCA1 and BRCA2 genes. The labeled DNA sticks to the probes that match it on the chip. Then the chip is scanned to see if there are any changes in the DNA. The scans show if there are any differences in the signals given off by the labeled DNA. Finally, doctors can compare the results to a database of known changes in the BRCA1 and BRCA2 genes. This helps them figure out if the person's DNA has any changes that could put them at higher risk for breast cancer. Using DNA chips can be really helpful for finding breast cancer early and figuring out the best way to treat it. But sometimes the results can be wrong, so doctors may need to do more tests to make sure they're correct. Next, let's talk about the importance of the BRCA1 and BRCA2 genes and how mutation in these genes can impact a person's risk of developing certain types of cancer. BRCA1 and BRCA2 are genes that produce proteins that help to prevent the growth of cancer cells in the body. However, when there are changes or mutations in these genes, it can increase the risk of developing breast, ovarian, and other types of cancer. Individuals who carry mutations in the BRCA1 and BRCA2 genes have a significantly higher lifetime risk of developing breast and ovarian cancer compared to the general population. For instance, women with mutations in these genes have a lifetime risk of up to 72% for breast cancer and up to 44% for ovarian cancer. Then, you may ask, what are the cons and pros when comparing DNA chip with traditional DNA detection methods? Before DNA chips were developed, scientists used techniques like northern blotting or quantitative PCR to study gene expression. These methods involve taking RNA from cells, converting it to cDNA through reverse transcription, amplifying it with PCR, and then separating the resulting products through gel electrophoresis or hybridization with labeled DNA probes. While these traditional methods provide useful insights into gene expression, they have a limited capacity for analyzing multiple genes simultaneously. DNA chips, on the other hand, allow researchers to analyze thousands of genes at once, making them a high-throughput approach to gene expression analysis. Despite the advantages of DNA chips, traditional methods like northern blotting and quantitative PCR are still widely used in genetics and molecular biology research. They're especially valuable for validating DNA chip results and studying specific genes or genetic variations in more detail. DNA chips are an amazing technology that allows researchers to study thousands of genes at once in a single experiment. This is called high-throughput analysis, and it provides a wealth of information that would be hard to obtain using traditional methods. Some of the great things about DNA chips include their speed, accuracy, and specificity. They also offer flexibility because they can be designed to analyze gene expression in any organism or tissue type. All of these features make DNA chips a powerful tool for studying gene expression and understanding the molecular basis of disease. There are several products and services that use DNA chips, which are small devices used to analyze genetic information. One popular service that uses DNA chips is 23andMe, where customers provide a saliva sample that is analyzed to identify genetic variants related to disease risk, ancestry, and physical traits. Another DNA chip-based platform is the Affymetrix gene chip, which is used to analyze gene expression in different species for disease diagnosis, drug discovery, and personalized medicine. The Illumina bead array is also a microarray platform that uses DNA chips to analyze gene expression and genetic variation, with similar applications in disease diagnosis, drug discovery, and genetic testing. GeneSight Psychotropic is a genetic test that uses DNA chips to analyze genetic variants associated with mental health conditions. This test helps clinicians identify the most effective psychiatric medications for their patient. Similarly, Veracyte Afirma is a diagnostic test that uses DNA chips to analyze genetic mutations associated with thyroid cancer. The test is used to distinguish between benign and malignant thyroid nodules, avoiding unnecessary surgeries.